So it's my great pleasure to introduce our graduate student speaker for this fall, Katherine Wood, who is co-advised by Dr. Kobe and Dr. Newman in the department. And I'll let her give you more about herself and introduce her exciting project today. Thank you, Katherine.
Yeah, thank you for the introduction. As she said, my name is Katherine Wood, and I am a PhD candidate in the Department of Forestry and the program in Ecology, Evolution, and Behavior at Michigan State University. And today I'm going to be presenting part of my dissertation research on how tree seedling functional traits mediate survival responses to light availability and plant soil feedback. Identifying the mechanisms that maintain species richness is a central question in plant ecology. In particular, we can understand how forest communities assemble by looking at tree seedling demography. The seedling life stage is a major demographic bottleneck and is thus a critical stage for the maintenance of forest community. Like adult trees, seedlings need to perform several basic functions, like maintain positive carbon balance, uptake water and nutrients, defend themselves against enemies, and store resources for the future. However, unlike adult trees, seedlings face several unique challenges. They're much smaller in size, they have far fewer resources, and much lower defenses. In thinking about the unique challenges that seedlings face, there are several mechanisms that have been proposed to explain why some seedlings survive while others don't. One of the most influential mechanisms proposed is plant soil feedback, which is the process by which plants alter the biotic and the abiotic qualities of the soil that they're growing in, which then alters the ability of plants to grow in that soil in the future. For example, an adult tree may modify the soil beneath its crown, thereby increasing or decreasing tree seedling survival in that space. And this can, in turn, affect which tree species will replace that adult in the future. If seedlings have higher survival in soil that's modified by adults of their own species, this is considered to be a positive plant soil feedback. And this can lead to dominance of a single species within a forest. Alternatively, if seedlings have higher survival in soil that's modified by adults of other species, this is considered to be a negative plant soil feedback. And this can serve to increase diversity within a forest by increasing the likelihood that a different species will replace an adult tree when it dies. The majority of plant soil feedback research has focused on soil-borne microbes as drivers. Specifically, research has investigated the roles of mycorrhizal fungi and pathogens. How tree seedlings respond to these microbes can shift with environmental factors like light availability. Mycorrhizae are typically mutualistic, and they can enhance seedling performance and high light availability when resources are more readily available. However, in low light availability, arbuscular mycorrhizal fungi may parasitize tree seedlings and exacerbate the negative effects of pathogens. On that note, soil-borne pathogens, which can cause the death of entire seedling cohorts, are typically more abundant in low light availability, where conditions enhance microbe reproduction and dispersal. Furthermore, the strength and direction of plant soil feedback differs between functional groups. Susceptibility to negative plant soil feedback increases with species rarity, decreases as seed size increases, decreases as shade tolerance increases, decreases and is higher for AMF than for EMF associating species. And we believe that functional traits may act as the mechanism underlying these patterns. Previous studies attempting to link functional traits to plant soil feedback have primarily focused on growth metrics like relative growth rate, specific leaf area, and specific root length. These studies often infer an inverse relationship between investment in growth and defense traits. However, growth defense trade-offs are not ubiquitous across species and can vary with resource availability. Furthermore, within the first growing season, impacts of disease often result in seedling death rather than decreases in growth. So to disentangle how functional traits mediate plant soil feedback, it's essential to quantify traits that directly confer defense or recovery against microbe-mediated mortality. Specifically, AMF compete with pathogens for space on roots, and EMF can form a physical protective sheath. Phenolics and lignin act as chemical and physical defenses against microbes, and non-structural carbohydrates can be mobilized to repair damaged tissue. We expect that defense and recovery traits should be expressed more in conspecific soils where mutualists accumulate and can provide more resources, and where specialist pathogens can also be more abundant and increase production of inducible defenses. Similarly, we expect that traits should be expressed more in high light where carbon is less limiting. In thinking about this, we came up with a couple of research questions. First, how does seedling functional trait expression vary in response to light availability and soil source? And second, how do tree seedling functional traits then mediate survival? And then underlying both of these questions, what explains these species-specific trait differences in trait expression and survival response? Can we generalize these to something like seed size or rarity or shade tolerance? To answer these questions, we conducted two experiments, a field experiment in which we utilized a common garden approach and a greenhouse experiment. In both of these, we manipulated soil source and light availability for our study species. The research site that we chose is located in a 100-hectare mixed hardwood forest stand in mid-Michigan at Alma College's Ecological Field Station. We identified adult trees for soil collection and established field plots in a 3-hectare mapped section of the forest where species ID, location, and size have been mapped for all species. We chose seedlings of 4 temperate tree species that naturally co-occur at this research site: Acer saccharum, commonly known as sugar maple; Prunus serotina, commonly known as black cherry; Quercus alba, or white oak; and Quercus rubra, or red oak. We chose these species because they not only co-occur within the same site, but they also differ in seed size, local adult abundance, shade tolerance, and mycorrhizal colonization. We chose to conduct parallel field and greenhouse studies because they present very different benefits and challenges to understanding plant soil feedback. For one, stressful field conditions can obscure the importance of functional traits for mediating survival. But on the other hand, plant soil feedback quantified in highly controlled greenhouse conditions often overestimates field measured plant soil feedback. In the field study, we took precautions against potential competition and above ground herbivory from rodents and deer. However, seedlings also experienced variability in rainfall and maximum temperature across the growing season. Drought that could result in seedling desiccation and rainfall that could wash away small individuals could have negated some of the effects of the traits. And similarly, variation in maximum temperature could have confounded effects of light availability. But results in the greenhouse aren't quite as meaningful as results in the field because they're in more controlled conditions. In addition to environmental stochasticity, the design of a field versus a greenhouse experiment to investigate plant soil feedback differ greatly. Soil placed in greenhouse pots doesn't act the same as soil in the forest, but collecting intact soil cores and attempting to limit confounding factors in the field is very time intensive. Similarly, although it is easier to create exact light conditions in the greenhouse using shade cloth on a greenhouse bench, it's more difficult to translate this to variation in light availability that would be experienced by seedlings in the field. However, in the field, it's also harder to control for bad weather events like heavy rainfall or temperature. So in these experiments, at 3 weeks, we harvested seedlings to quantify defense and recovery functional traits of interest. Specifically, we measured colonization by AMF, colonization by EMF, phenolics, lignin, and nonstructural carbohydrates. We also monitored our seedlings for survival over the first growing season. To answer the first question, how does seedling functional trait expression vary in response to light availability and soil source? We looked at these functional traits under each light level and in conspecific versus heterospecific soil. In the field, we found that AMF colonization was higher in heterospecific than conspecific soils for most of our measured species. This was in contrast to other studies that have found that AMF colonization is higher in conspecific soil, where one might expect that specialist mutualists have had more time to accumulate. However, AMF are generalists, and they can associate with multiple tree species and even across genera. When we looked at individual soil sources, we found that AMF colonization was highest in 2 soils, Populus grandidentata, and Acer saccharum, suggesting that AMF from these soils can readily colonize multiple seedling species. Populus grandidentata is an early successional species and has some of the oldest individuals in the experimental forest. As such, AMF associating with Populus grandidentata soils have likely had the longest amount of time to establish beneath the crowns of these adults, and perhaps have even had time to acclimate to colonizing other species. Similarly, Acer saccharum is the most abundant species and has some of the largest individuals in this experimental forest. Like for Populus grandidentata, AMF associating with Acer saccharum soils are likely prolific within the forest and could explain the high amount of colonization that we see in these soils. Consistent with our expectations, EMF colonization was higher in conspecific than heterospecific soil. And this was expected because EMF is more specialized than AMF, often associating with a single genus or single species. When we looked at individual soil sources for EMF, we found that for Quercus alba, EMF colonization was highest in soils cultured by 3 species: Populus grandidentata, Quercus alba, and Quercus rubra, species that all associate with EMF as adults. And this result suggests that Quercus alba seedlings can actually associate with multiple kinds of EMF and may have a more general relationship rather than the specialized one that we've come to know. For Quercus rubra, EMF colonization was highest in conspecific soil, especially at high light, but it was actually lowest in conspecific soil at low light, perhaps signifying a more specialized relationship that's more sensitive to resource availability. When we looked at phenolics, we found that phenolics were higher in conspecific than heterospecific soils for Acer saccharum and Quercus alba across light levels, and for Quercus rubra at high light. Phenolics production can be induced in response to soil-borne microbes, which should be more prevalent in conspecific soil. Phenolics were also, did I switch the slide? Okay, no. Phenolics were also sensitive to light, increasing with light availability for all of the study species. And we didn't quantify pathogen abundance in this study, although we expected that higher pathogen abundance would likely drive higher phenolics production. But we did find that for the 2 oak species, Quercus alba and Quercus rubra, EMF colonization and phenolics were correlated. Since EMF colonization was also higher in conspecific soil for both species, we expect that EMF colonization induced production of these phenolics, especially in conspecific soils. For Prunus serotina, phenolics were higher in heterospecific soil, and they were positively correlated with AMF colonization. Prunus serotina seedlings may be more readily colonized by generalist AMF, regardless of soil source, and thus produce more phenolics in response. Lignin expression was strongly associated with seedling size and mycorrhizal association. For the large-seeded EMF associating species, lignin was higher in conspecific than in heterospecific soils, but it didn't vary with light. In conspecific soil, when mutualistic EMF are available, seedlings may have more resources available for lignification. Furthermore, large-seeded species have more maternal reserves to allocate to defense traits. So lignin production may be less responsive to light or plateau at a threshold value when carbon from maternal reserves is already high. For the smaller seeded AMF-associated species, Acer saccharum and Prunus serotina, lignin did increase with light, but it didn't vary with soil source. So we concluded that small seeded species rely more heavily upon resource availability in the environment, and they may not be able to produce as much lignin at low light without those maternal reserves. And finally, non-structural carbohydrates increase with light availability for all of our species, suggesting that non-structural carbohydrates are highly responsive to light, regardless of soil source. But contrary to our initial expectations, they were higher in heterospecific soil across all light levels for Quercus alba and at low light for Quercus rubra. In conspecific soils or at low light availability, seedlings may allocate more non-structural carbohydrates to their mycorrhizal mutualists or to recovery from pathogens. However, we can't necessarily conclude that this was the case because we didn't find strong correlations with non-structural carbohydrates for either AMF or EMF. So in summary for question 1, our results clearly demonstrated that tree seedlings, even as young as only 3 weeks old, express intraspecific variation in functional traits as a response to light availability and soil source. And there was significant variation in species responses, but we were able to make a handful of generalizations. For AMF colonization, there may be higher colonization for shade intolerant species and in soils that have been cultured by older or more abundant adults in the forest. EMF colonization, which is generally thought of as a more specialized relationship, may be specialized for some species, but more general for others. Phenolics increased in conspecific soil, which aligns with our expectation that it's an induced defense, and also increased with light availability. Lignin was dependent upon seed size and mycorrhizal association, generally increasing in conspecific soil. And non-structural carbohydrates were higher in heterospecific soil, likely because they're expended in conspecific soil and also increased with light availability. So once we had determined that these traits do vary in response to light and soil, we asked ourselves, how do these traits mediate survival? What we found was that species differed in which traits, if any, influence their survival. We found that Acer saccharum seedlings had higher survival in sterile than non-sterile conspecific soils, and they also had slightly higher survival rates in conspecific than heterospecific soil, suggesting a microbially mediated plant-soil feedback. Our data suggests that phenolics, which provide direct chemical defense against soil-borne microbes, increase survival for Acer saccharum seedlings. Host-specific microbes accumulate in conspecific soils, which then induces greater production of phenolics. Acer saccharum seedlings had higher levels of phenolics and experienced greater rates of survival in conspecific soils, suggesting that there was a positive feedback driven by this chemical defense production. Furthermore, Acer saccharum had higher survival in sterile soil, confirming that microbes were driving this effect. In the field, we found that Quercus alba didn't show strong differences in survival due to soil source, but survival decreased as AMF colonization increased. EMF-associated species typically experience positive plant-soil feedback, meaning that they have better performance in conspecific than in heterospecific soils, whereas AMF-associated species typically exhibit negative plant-soil feedback. EMF may provide better direct defense against pathogens, while AMF can act parasitically under certain environmental conditions. However, it's not well understood if AMF caused negative plant soil feedback for EMF associating tree species. A likely reason for the increase in Quercus alba seedling mortality with AMF is actually the lack of a common mycorrhizal network of EMF. In forest ecosystems, EMF mycelia can link roots of neighboring trees and seedlings, thus facilitating transfer of resources between individuals. The design of our field and greenhouse pots inhibited mycorrhizal hyphae from passing in or out of the soil, limiting seedlings to colonization by microbes that were present during initial soil collection. Quercus alba seedlings had lower colonization by AMF and higher colonization by EMF in conspecific soils, supporting that they are preferentially colonized by EMF as seedlings. Furthermore, they had more colonization by EMF in soils that were cultured by other EMF-associating tree species, indicating that they prefer when more EMF are available for colonization in these soils. Similar to the field experiment, in our parallel greenhouse experiment, we also found that traits predicted survival, but the results were, again, very species-specific. For Prunus serotina, survival was higher in sterile than in non-sterile conspecific soils. We also found that increasing AMF colonization decreased survival. We suspect that as colonization of AMF increased, allocation of carbon to AMF also increased, and seedlings had less resources available to recover from disease. In this scenario, AMF are acting parasitically and can exacerbate the negative effects of pathogens. Previous research has demonstrated that some plant species are able to regulate the amount of AMF that can colonize their roots and how much carbon is allocated to them. But it appears that Prunus serotina maybe can't regulate this relationship as much as other species and suffers higher mortality for it. We also found that phenolics increase survival for Prunus serotina seedlings. Since phenolics act as an induced defense, we expected to see higher production of phenolics in conspecific soils and when more microbes like AMF were present. It may be that as AMF act parasitically and reduce seedling survival, increased production of phenolics serves to counteract this. So seedlings utilize a chemical defense against pathogens rather than allocating carbon after attack. In the greenhouse for Quercus rubra, there really wasn't much variation in survival due to soil source, although it appears that the seedlings have slightly higher survival in conspecific soil, regardless of sterilization. We found that EMF colonization increased survival for Quercus rubra. EMF, unlike AMF, have not been shown to act parasitically. They have more specialized relationships with their hosts, and they can create a physical barrier on root tips, providing direct physical defense against pathogens. And this may explain why survival in conspecific soil didn't differ from survival in sterile soil. Although more harmful pathogens are present in the non-sterilized conspecific soil, the presence and colonization of EMF may protect seedlings and negate the presence of those pathogens. And if you recall from the field experiment results, we found that Quercus rubra seemed to have a more specialized relationship with its EMF that relied more heavily upon light availability. In the greenhouse, we didn't find strong differences in EMF colonization due to soil source, but we were still able to see an effect on survival, thus demonstrating how different plant soil feedback can be in the field versus in the greenhouse. And for Quercus rubra, we also found that lignin increased survival. Interestingly, there was no difference in lignin production in response to light availability or to soil source. Quercus rubra may allocate a fixed amount of its resources to lignin regardless of resource availability or microbe presence. And in that case, when pathogens are present, lignin is then present to defend against attack. And we expected to see traits emerge as stronger drivers of seedling survival, given the large amount of intraspecific trait variation that we saw in response to soil source and light availability. However, we were able to make some generalizations. I will note that in this summary table, I'm including results from another greenhouse experiment shaded in green. In that experiment, we investigated the role of shade tolerance for plant soil feedback. And so I present the results for Acer negundo and Acer saccharum in this table here. In the experiment, we found that Acer negundo, which is highly shade intolerant, had the highest amount of AMF colonization and also had increased survival as AMF colonization increased. However, in our parallel field and greenhouse experiments, we found that AMF colonization had a negative effect on survival, even for Prunus serotina, which is also a shade intolerant species. So this leads us to believe that relationships with AMF are variable and that there are more factors underlying the relationship beyond shade tolerance or seed size alone. As suggested before, maybe some species are better at regulating how much mycorrhizae can colonize the roots and how much carbon is allocated to them. We didn't really find a generalizable relationship between EMF and survival. However, we did find that EMF increased survival for Quercus rubra, which appeared to have a more specialized relationship with its mycorrhizae than Quercus alba. And perhaps the relationship is more specialized because EMF confer defense against pathogens for these seedlings. For phenolics, we found that they increased survival for 2 of our 3 small-seeded and AMF-associating species. We found that for both of these species, phenolics were higher in conspecific soils and were induced by microbe presence. These species also had negative relationships with AMF in survival, suggesting that chemical defenses may be in response to or even offset some of the negative effects of the symbiosis. And finally, lignin increased survival for Acer saccharum and Quercus rubra. These species differ in seed size and in mycorrhizal association, but they're both shade tolerant, suggesting that there might be some relationship to the growth defense trade-off hypothesis, which posits that shade tolerant species forego high light growth for low light survival. We found no direct effect of non-structural carbohydrates on survival in any of our studies, but we did find that non-structural carbohydrates tended to decrease as AMF increased, suggesting that there is an indirect relationship mediated by AMF colonization. Although there's increasing research attempting to link plant traits and environmental conditions to plant soil feedback, the majority of studies have focused on herbaceous plants, and they haven't investigated the role of functional traits that provide direct defense against microbes. A focus on tree seedling traits under different environmental conditions, like light availability, offers both broader ecological understanding, as well as potential application for forest management. For example, selecting sites with soil and light conditions that could promote higher production of defense compounds could increase the likelihood of seedling restoration success. Similarly, it might be beneficial to plant EMF associating species in soils that are cultured by other EMF associating adults to increase the potential for positive effects of a common mycorrhizal network. While variable environmental conditions can obscure trait effects on seedling survival, a sharper focus on traits promoting survival rather than growth in comparisons of field versus greenhouse studies will provide a more mechanistic understanding of forest community dynamics. Before questions, I would like to acknowledge the people who have contributed to this study. First of all, my advisors, Rich Kobe and Sarah McCarthy Newman, and then my three other committee members, David Rothstein, Mike Walters, and Andrew Jarris, and then the various people who have contributed to the success of the field study in particular, and to helping with the revisions and data analyses. Does anyone have any questions?
Hi, Katherine. It was a really good talk, and I'm running the experiment myself, and using the red oak and white oak, and I'm very curious about the different result between the two oak species, specifically the different slopes in the light response. And did you find any literature that emphasize the some kind of difference between the two oak species that's co-occur in this region, like physiological or some kind of traits that can make them different in response to light?
Yeah, I didn't really find any literature that showed that. I believe it's Quercus alba that is slightly less shade tolerant than Quercus rubra, if I'm recalling correctly. So that might have something to do with the response to light. But no, I didn't find that. I do have some results from nutrient analyses that we did where there were differences in the response to different nutrients. And I'd be happy to share that with you later if you want.
Yeah, I'm curious about that. And I have another question. So you manipulated the light environment, right?
Yes.
And did you see any effect on the temperature to the seedlings co-bearing with the the shade manipulation?
Yeah, so we actually found that the maximum temperature reached in the field plots had a significant effect on tree seedling survival. And so it confounded some of the effects of the light availability, but we kind of offset that by also doing the same experiment in the greenhouse where the temperatures were more controlled.
Thank you. Interesting.
There's a question in the chat from Georgios. It says, I have a question about shade tolerance traits that you mentioned. Did you explicitly include shade tolerance in the modeled relationships? And if yes, was it confounded with other functional traits? We actually did not explicitly use shade tolerance in the modeled relationships. When we looked at survival, we ran 2 separate models. The first one was using environmental variables, light and soil. And in the field experiment, we also looked at maximum temperature and soil moisture. And then our second survival model just used the traits. There was a lot of covariation between the traits and then also with those environmental variables. So it's really hard to include all of them in the same model.
Katherine, I have another question. So you harvested seedling at the after 3 weeks of growth.
Yes.
And do you think that if you have harvested the seedling, say like 6 weeks after the growth, would you expect to find different results in the measurement?
Yeah, so we actually looked at some of the traits later on towards the end of the growing season. And then for the greenhouse study, we actually collected traits not only at 3 weeks, but then also at, I believe it was 12 weeks. And we didn't find that the relationships with survival changed much with the increase in age. And the reason why we picked the 3-week-old trait specifically is because in previous studies, we've seen that peak seedling mortality occurs between 3 and 9 weeks. And so we were really interested to see if traits right below that peak mortality could kind of predict that. Yeah.
Yeah, interesting. Thank you.
Are there any other questions?
Again for a great presentation, and thank you to everyone for attending. And we'll see you all again next spring.
Thank you for coming, everyone.